Hi guys, we are back with prokaryotic cell division. Alright, when we talk about a prokaryotic cell, if you remember what I said, pro means before. Karyon means nucleus. So these are very primitive forms, bacterial forms, that do not have a nuclear membrane. Very, very simple. They have cell wall, they have a cell membrane, they have cytoplasm, they have chromosomes, and of course the usual ribosomes, which we had talked about in the previous slide. I am not going to talk about it right now. They do not have a nuclear membrane. They have a single circular chromosome. So single circular chromosome on which you find DNA. This is obviously old information. So how does prokaryotic cell divide? How does a bacterial cell divide? Well, there will be definitely one term that you'll come across, asexual. A means no. So here, this reproduction does not require two parents. So here, this requires only a single parent. A single parent is enough. A single parent will do it, okay? Single parent. Two parents are not involved. And of course, the offsprings are genetically identical. So all the babies are going to be exactly like the parent, like the mother. All right? Another word that you'll come across is binary fission. Bi means two. Fission means breakdown. So simply what it means is that a prokaryotic cell divides and produces two genetically identical daughter cells by cell division. Okay? So one will divide into two. Fission means breakdown, bi means two. Alright, so first and foremost we have the prokaryotic cell. And the prokaryotic cell, what we see is that the single circular chromosome has replicated. So now there are two chromosomes, two single circular chromosomes, and they're attached on the inside of the cell membrane, if you can see this, right? All right, now here what we see is that the two chromosomes, now the cell membrane that is right there, where these are attached, this area starts becoming stretched. And as it stretches, so here we have one chromosome hanging and we have the second chromosome hanging, this region starts becoming stretched. And as it becomes stretched, the two single circular chromosomes, these will start separating. All right? As they start separating, you will notice that the cell membrane and the cell wall, this starts to grow inwards. And as it grows inwards, you're noticing that the cell is stretching, elongating, cell wall material, cell membrane material is being deposited, and the chromosomes are becoming separated, more and more separated, and they are going towards the opposite ends. Once the cell is approximately twice the size, initial size, original length, then what happens is the new cell wall and cell membrane division is complete. It grows inwards, the cell gets pinched, and once it gets pinched, then you will notice that two daughter cells are formed. Now the E. coli, the cell division completes within 20 minutes, very, very fast. So in 7 hours, you can go from one cell into 1 million cells. But there are other bacterial cells that are slower. They may take up to 24 hours to complete cell division. And this you can talk about, we'll talk about this later in higher classes. And so this is a very short presentation on prokaryotic cell division. I hope this was helpful. I thank you for your time. If there are any questions or concerns, please stop by my office or call me or talk to me. Thank you.